Hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Students, quickly let me know in the chat right now, before I start today's amazing class, if I'm properly audible and visible to each and every one of you students. Quickly let me know in the chat if I'm properly audible and visible. So students, this is a very, what do I say, fundamental chapter, right? So if you want to understand the chapters of your class 11th, plant physiology and human physiology, or the chapters of your class 12th, molecular basis, genetics, human reproduction, plant reproduction, this is the foundation chapter. Unless you understand this chapter, you cannot go further. You cannot go further, students. So if you are ready, students, if you are ready for today's class, quickly fire emoji in the chat, all of you students. Quickly fire emoji in the chat to let me know, sir, yes, I am ready for today's class. I am going to learn a lot of things. Now, first of all, students, who should watch this lecture? A student who has zero understanding, or the student who is preparing for NEET 2026-27 from the scratch. You have zero knowledge, you can watch this. If you do not know anything about cell, you can watch this. If you know something, well and good, because 90% of learning will happen in the class. You will write the notes with me, you will learn with me. Okay, students, all of you ready? Students, in the chat quickly. All of you ready? All of you in the chat quickly. Can I get a fire emoji, students? Can I get a fire emoji in the chat? Yes. Now. Let me ask all of you a very simple question. Very simple question. Students, a simple rule in my class. Very simple rule throughout the year. Every single time I ask you a question in my class, everyone should spam the answer in the chat. Deal, students? Deal, all of you? So every single time I ask you a question, you should completely spam the answer in the chat. That is my simple rule in my class. Okay, I want you to be active. Now I want all of you to look around yourself and tell me three things which you see around yourself. Three things which you see around yourself. For example, in my case, I see lights, I see camera, and I see all of you, right? There is a board here, right? Then I see certain books lying around. Then I see my gaming console there. I can see my dog outside. He's sleeping outside, right? So I see a lot of things around me. I see lots and lots of things around me, students. Now in this entire things, everything can be divided into two categories. Your brother also, okay, your brother also, okay. All of these things can be divided into two categories. Let's say it's Varsha's brother. Let's Varshita, right? Varshita's brother is a living organism. Let's say in Harini's room there is one cockroach. Don't tell her, but okay, there is a cockroach. Cockroach is living. In, let's say, in Rushti's house there is a spider in the corner. That is a living. Some of you have pen stand, books. That is non-living. So around you right now, you can see something called as, students. You can see, all of you can see, students. All of you can see something called as living as well as non-living things. Yes or no, students? Around all of you students right now, you can see something called as living things as well as non-living things. Now you might wonder, what is the difference, students? What is the difference between living things and non-living things? What is that fundamental difference? What is that fundamental difference between living and non-living? Very simple, students, very simple. All of these living things are made up of a tiny structure called as dash. All of you in the chat. All the living things are made up of a tiny structure called as cell. Okay, they are called as cells. Now students, all of you might have seen a house building, right? All of you might have seen this houses. All of you also have a house. How is the house made, students? The house is made up of tiny bricks. Yes, this entire house is made up of tiny bricks. Yes, similarly we can use Legos to make one entire Lego building. Yes, similarly students, I want all of you to understand our entire body, you, me, all of you, all the entire body, students, is made up of cells. Now I will show all of you one small image which all of you will remember. You might have done this experiment, you might have not done this experiment, called as onion peel experiment, students. How many of you have done this experiment? How many? Raise your hands, students. Okay, this is an onion peel here. In this onion peel, can all of you see It is made up of tiny brick-like structures. Tiny brick-like structures called as cells. It is made up of cells, right? It is made up of cells. Now, one ultimate question to all of you. Ready? Varshita, Harini, Achu, Roshni, yes, Manani, all of you ready? One ultimate question. Okay. Do you think an atom is smaller, or do you think a cell is smaller? In the chat, go. A simple question, yet also tricky. It's not tricky actually. It is a very basic question which many students sometimes do not know. Okay. So what is smaller? All of you tell me, what is smaller? Okay. Do you think a cell is smaller, or do you think an atom is smaller? So if you have basic understanding of physics and everything, you will tell me, sir, atom is smaller always. Okay. Atom is always smaller. So atoms will make up molecules. Molecules will make up the cell. Students, molecules will make up the cell. Now, when we have multiple cells, and we join everything together, what do we get? When we join multiple cells, we get tissue. Yes or no, students? So from a cell, we get tissue to organ to organ system, and organ system to a organism. This all of you should know, students. This all of you should know. Now, students, coming back to our introduction to cell here, I want all of you to understand every single life on earth starts with a single cell. I repeat, students, every single life on earth starts with a single cell. Every life starts with a single cell. Now, students, can anyone tell me, in the case of humans, what is a cell called as? In the case of humans, the cell is called as what? It is called as zygote. It is called as zygote, students. Now, this zygote divides, and it will make up the trillions of cells. So, this single zygote can divide, and we can get trillions of cells. One single zygote. Now, students, do you think there is some organisms in the world? They are starting from a single life. They are starting from a single cell, but they never divide only. Students, do you think it is possible that an organism can survive with a single cell? Yes, it is possible. So, students, I want all of you to understand that this living organisms, right, they can be of two types. They can be of two types here, which are made up of a single cell. Some are made up of many cells. Okay. If they are made up of a single cell, it is called as unicellular organisms. They are called as what? Unicellular organisms. What about many cells? They are called as multicellular organisms. So we have unicellular organisms as well as your multicellular organisms. Clear, all of you students? Clear, Darshini? Yes? Clear, all of you? Now, students. Now, I want all of you to give me a quick example. See, unicellular organisms, all of you know examples. Bacteria, amoeba, paramecium. Now, can anyone give me the examples of multicellular in nature? Anyone? Multicellular organisms. Anyone? Quickly, students. Quickly, all of you in the chat. Quickly in the chat, students. Anyone? Any examples of multicellular in nature? You will tell me and you, students. Me and you. We are humans. Then do you think plants? Plants. What about, students, animals? Animals also. Students, what about fungi? What about fungi, students? Apart from yeast, all of them are multicellular in nature. Apart from yeast, they are multicellular in nature. Now, but students, I am talking about cell this, cell that, uni, multi. But what is the definition of cell? Can anyone in the chat from your class 10th, 9th, 8th, 7th, 6th, what have you learned a definition of a cell, students? Definition of a cell. So what exactly is the definition of a cell? You will tell me, sir, cell is nothing but structural. It is nothing but structural and functional unit of life. Yes or no, students? Yes or no? This is the definition taught you, students. This is the definition taught to you. Yes. So I will just modify it a little. I'll just modify this definition for the students. Okay. Cell is the basic structure. Cell is the basic cell is the basic structure and function unit of life. Okay, now I want all of you to understand it is actually the smallest unit. Just try to imagine this is the smallest unit. Okay, it is capable of independent existence. Independent existence, students. It is able to independent existence, students. It is able to preserve. It is capable of independent existence. Independent existence. Now, what do I mean by that, students? What do I mean by independent existence? This one cell. Now, try to imagine one BHK apartment. Okay, there is only one apartment here. Okay, in one apartment, to survive in your house, what and all do you need, students? Let's say there is one house. Okay, in one house, what and all do you need? Do you need a main living room? Okay, we have a living room. We have, let's say, if you have little money, you have dining room is there. Okay, then you need a kitchen to cook the food. Okay, then you need something called as what? Bathroom to remove the waste. Okay, then if you have more money, balcony and all that is there. Similarly, students, how we have one house, right? Similarly, cell also is like that. One cell is like a small room. It can do every single function required for our body. For example, a cell should be able to do respiration. A cell should be able to do reproduction. A cell should be able to do excretion. So every single students, every single function can be done by a small unit called as a cell. Small unit called as a cell. Now this is especially, students, this is especially true for unicellular organisms. In the case of unicellular organisms, right, it can show independent existence, and there is no division of labor here, students. There is no division of labor here. No division of labor at all in the case of unicellular organisms. That one cell is only doing everything. One cell only is doing every single function there. Okay. Now, I want all of you to understand anything less than a complete structure of a cell is not cell at all. Okay, it will not show independent existence only. Now try to imagine there is no bathroom in your house. Can you survive there? All of you tell me yes or no in the chat. In your house there is no bathroom only. Can you survive? You have to go outside somewhere. But again, you will not be able to live inside. Let's say in your house there is no kitchen only. Will you be able to survive? No. So anything, if a house is incomplete, right, you cannot live there. Similarly, if in a cell something is not there, you cannot. The cell is incomplete. Okay, the cell is incomplete, students. Now, a question to all of you, students. A question to all of you, students. Okay, who discovered the cell? Who discovered the cell, students? Who discovered the cell? You might know this. All of you might know this. Okay, let me rephrase the question. Who discovered the dead cell first of all? Anyone, students, who can tell me? Who discovered the dead cell first of all, students? Who discovered the dead cell? Anyone who can tell me, students? This scientist here, this handsome sir here. Who discovered the dead cell first of all? The dead cell was first time discovered by a scientist called as Robert Hooke. Okay, this right here, students, is your Robert Hooke. Robert Hooke. Now, but I want all of you to understand. Do you think Robert Hooke was the first scientist to use the microscope? Students, do you think that? Do you think Robert Hooke was the first person to use this microscope? No. There was Janssen. Then there was Galileo. After Galileo, we have Robert Hooke. After Robert Hooke, we have Antony von Leeuwenhoek. Okay. So Robert Hooke was the first scientist to observe the dead cells. Now, I want all of you to understand, what do you mean by dead cell? Okay. Now, do all of you know what is the meaning of protoplasm? Does anyone know? What is the meaning of protoplasm? Protoplast? Do all of you know? Should I teach? Students, ask me. See, in my class, you can ask. You can ask doubts and everything. Do all of you know what is a protoplasm? Yes or no in the chat. Okay. Because to understand the Robert Hooke's work, you need to understand what do you mean by the protoplasm. Listen to me, students. Listen to me. Okay. Now, let's say this is one cell right here. This cell right here. Okay. Let's say I have a cell here. Let's say I have a cell here. Okay. Now, this cell right now, students, this cell right now has something called as cytoplasm. Cytoplasm is different. Cytoplasm is different. So inside we have the cytoplasm. Cytoplasm. Okay. Now apart from cytoplasm, students, there is something called as, right in the middle, there is something called as nucleus also. Nucleus also. Then there is mitochondria here. Then there are other different organelles here. Everything is there. Now this is called a cytoplasm. Now students, whatever is present inside the cell membrane, whatever is present inside the cell membrane, inside here, this inner part here, that is basically the cytoplasm plus nucleus plus other organelles. This entire thing together is called as what? It is called as protoplasm. So whatever we have inside the cell membrane, whatever is present inside is called as your protoplasm, students. Protoplasm. And this protoplasm, students, is actually the living part of the cell. It is actually the living part of cell. Students, is that clear to all of you? Protoplasm is clear to all of you, students? Cytoplasm plus nucleus plus other organelles are part of your protoplasm. Now, I want all of you to understand, what did actually Robert Hooke discover? Okay, he actually never discovered the cell. He never observed the cell actually. Robert Hooke never observed the cell. Very important, students. Okay, Robert Hooke actually observed something called as cork cells. He observed something called as cork cells. Understand what is cork, students? Understand what is cork cells, students. Kanul understand the meaning of protoplasm. The meaning of protoplasm is cytoplasm plus the nucleus plus other organelles. All three together is called as protoplasm. Okay. Now, what do you mean by cork cell, students? What do you mean by cork cell? Listen to me, students. This is a tree here. It has a bark outside, bark. This bark has a outer layer called as cork. That is the bark only. Okay. And when he observed this is what he observed. Students. Now I want all of you to understand, students. Let's take a plant cell here. Let's take a plant cell. Okay. Let's say this is a plant cell right now, and this plant cell right now has a cell membrane on the outside. Okay. This plant cell has a cell membrane here. Okay. Now, students, once this particular cell dies, after it dies, what do we have, students? Once this particular cell dies, the remaining part is going to be the cell wall. Yes or no? Only the cell wall will be remaining here. The inner part, the inside, is completely hollow. It is completely empty. On the inside. This is what was observed by Robert Hooke. Robert Hooke observed this one, student. This is what Robert Hooke observed, students. Okay. So Robert Hooke observed, right? He observed, students. He observed the cell wall. Actually, he observed the cell wall. Okay. So he observed the cell wall enclosing the protoplasm. So he observed the cell wall. He actually did not observe the cell at all. Cell was gone. Cell was dead already. So he observed a hollow structures. Okay. So students, tell me, this hollow structures, right? These hollow structures, don't they look like honeycomb? Tell me, yes or no, students? Don't they look like honeycomb here? All of you in the chat who can tell me. Students, don't you think so? They look like honeycomb here? Yes or no, students? Honeycomb. They look like honeycomb, right? Yes, they look like honeycomb. Now he called these honeycomb structures as small compartment. Okay, he called this honeycomb as small compartment. Small compartment or small space. And this small compartment, in his language, Latin, it is called as, students, it is called as what? Cellulae. It is called as cellulae, student. It's called as cellulae. And today we know it as this cellulae, student. This cellulae, today we know it as cell. That is how cell came into existence by Robert Hooke. Robert Hooke. He observed the cork cells which were dead. He only observed, students, he only observed the cell wall here in like a honeycomb structure. They look like a small compartments or small rooms here, tiny rooms, and he called them cellulae, and we call them cell in today's world. We call them cell, and that is how, student, that is how the existence of cell was first time came into picture. Students, how many of you understood? If you understood, a quick fire emoji in the chat, let me know that yes, you're understanding the first and foremost the history of cell. Students, all of you understood? Students, all of you understood? Yes or no? Students, all of you understood? Yes. This is the story of Robert Hooke. This is the story of Robert Hooke here. Okay. Now, then came second scientist called as Antony von Leeuwenhoek. Antony von Leeuwenhoek, students. So Antony von Leeuwenhoek was a scientist who observed the living cells. He observed the living cells. Living cells. Now you might ask me, sir, which living cell did he observe, students? Which living cell did he observe? Very simple, students. The living cell observed by Antony von Leeuwenhoek was none other than the bacteria, spermatozoa, right? So he observed the bacteria, protozoa, right, spermatozoa, and RBC, not RCB, RBC. RBC is called as red blood cells. Now, even he observed water. In water only he found all this. Like imagine he's going on the road, nicely walking on the road, okay? Then he sees some water there. Take and observe. Now, students, now you only tell me, when you observe these microbes, do you think they look like one small animals moving in water? Yes or no, all of you? Roshni, Rusty, Subramanian, Aishwarya. Students, I can't see my specs. I can't forget. I don't know where I kept my specs today. Okay, so they look like small, tiny animals moving in water. Yes. So he actually observed, students. He actually observed something called as, right? He called them, students. He found something called as little animals, and this little animals, he called them, students. He called them animalcules. He called them animalcules. Small, tiny motile structures. He called them animalcules. Animalcules came into existence. So that's what is observed by your Antony von Leeuwenhoek, students. Okay. Now, next scientist is called as your brown person here, called as Robert Brown. Robert Brown discovered the nucleus. He discovered the nucleus in the case of orchid roots. In the case of, students, orchid roots, students. Okay, in the case of orchid roots, he observed the nucleus, Robert Brown. Now, students, after this there was electron more microscopy developed, and we found other organelles one by one. Okay, students, how do you think till now all of you understood? If I can get a red hearts in the chat, will tell me yes, all of you understood the history till now. Because the next step is very important, students. The next step which we'll be going is called as cell theory, students. Cell theory. All of you ready, students? All of you ready? All of you drink some water, students. All of you keep always water bottle next to you. Always keep a water bottle next to you, students. And students, quickly smash a like button, all of you. Quickly smash a like button right now, all of you. Smash. Okay. Now, let's go to the next cell theory right now, students. Let's go to cell theory right now. Okay, shall we start, all of you? Shall we start? Cell theory. Shall we start? Yes. Now, cell theory is very important, students, because there are three main scientists here who are part of cell theory. In old NCERT, it says one was British, one was German. Now, both are German right now. There are two main scientist students here. One is called as Scheiden, one is called as Swan. Okay, two main scientist students here, two main scientists. Okay, in 1838 and 1839, okay, two scientists gave a major contribution to the cell theory, students, cell theory. The first scientist is called as Mathis Scheiden. Okay, Mathis Scheiden here. Okay, Mathis Scheiden. Mathis Scheiden, students, was a botanist like me. He was a botanist. We'll write bot. Okay, botanist. The one more scientist in 1839 was called as Theodore Swan. Students, just see the name. It's a beautiful name, right? Beautiful name, Theodore Swan. Okay, Theodore Swan. Theodore, like some movie name it is. Okay, Theodore Swan. He was a zoologist. He was a zoologist, like Gopika ma'am. Okay, like a Gopika ma'am. Now, students, I want all of you to understand. Next part will come. Don't worry, students. Why you see? Focus on first part first of all. Now, all of you tell me in the chat right now. This entire his name, your Mathis Scheiden. He is a botanist. Will he work on plants or will he work on animals? You tell me in the chat, students. Obviously, students, he'll work on plants. So his main work was on plants, and he told us that, students. He told us that all the plants are made up of different kinds of cells. Can anyone tell me the different kinds of cells we find here, students? Different kinds of cells. For example, you know there is something called as your meristematic tissue, right? Meristematic cells, right? Then all of these cells, they make up something called as tissue eventually. Tissue. We have something called as your collenchyma, parenchyma, sclerenchyma, xylem, phloem. So there are different types of tissues found in the case of your plants. Now, your Theodore Swan told us, students, he told us that Theodore Swan told us that here in this case, in the case of animal cell, in the case of Theodore Swan told us that in the case of animal cell, it has a covering. And this covering, today we know it as cell membrane or plasma membrane. So he told animal cell has a covering. It is called a cell membrane. It is present even in the case of plant cell also. He told us that eventually. Now, he also told us, students, that there is a structure called a cell wall, which is very unique to your plant cell also. There is something called as cell wall, which is unique to the plant cell. Obviously, cell wall is present only in the case of plant cell. And at the finally, he told us that all the plants and animals. Again, students, this last statement, right? This last statement was even told by Mathis Scheiden also. All the plants and animals are made up of cells and products of cells. That is what told by your Theodore Swan. And this right here is actually the first law of your first part of your cell theory. Students. Okay. So see here, we have Mathis Scheiden here, who was a botanist, who told us that plants are made up of cells, different kinds of cells and tissues. Theodore Swan told us that animal cells has a covering. Animal cell has a covering around it, and that covering is called as cell membrane or plasma membrane. And he told us that this something called as cell wall. This cell wall is very unique to only to your plant cell. It is not found in the case of animal cell at all. Not found at all. Okay. And then, students, he told us that plants and animals are made up of cells, like we are made up of cells, and product of cells. Now you will ask me, students, what do I mean by product here, students? What do I mean by product here? Product means we have proteins, we have carbohydrates, we have lipids, different proteins, structures are there. All of these are called as product of cells. So we are made up of carbohydrates, we are made up of proteins, we are made up of lipids, and all the things. That was the main theorem about your cell theory. Now, all this is okay. All this is okay. But these two scientists could not explain how cells came into existence. Like, from where are the cells even coming from? Like, how do the cells come into existence? To tell this one line, how cells come into existence, there was one more scientist called as Rudolf Virchow. There was one more scientist, students, called as Rudolf Virchow. He was like, all of you be quiet. I know, I will tell you the answer. Okay, so Rudolf Virchow came and told us that cells exist from pre-existing cells. Students, here, this is your Mathis Scheiden and Theodore Swan here. Okay, this is Rudolf Virchow. Rudolf Virchow told us that, students, cells divide and cells, right, cells divide, okay, cells will divide, students, and new cells, right, new cells are formed from pre-existing cells. Okay, so there are some old cells are there, right? There are old cells which will divide and we get new cells here. Okay, so there are cells divide and new cells are formed from the pre-existing cells. That is what told by Rudolf Virchow. Now he also given Harry Potter spell here. Okay, also gave us Harry Potter spell. The Harry Potter spell is called, students, omnis cellula e cellula. Omnis cellula e cellula. This was given by Rudolf Virchow, students. Rudolf Virchow. Okay, so students, any doubt here? All of you, any doubt, students? Any doubt here? Students, I can't see the chat there properly. I'll see in my phone itself. Students, any doubt here? All of you. All of you. With this, cell theory came into existence. Okay, so cell theory mainly has two things actually. According to your NCERT, there's only two things. According to NCERT, right, cell theory has only two things. Plants and animals are made up of cells and product of cells. First point. And second point, students, is this one. Cells divide and new cells are formed from the pre-existing cells. Now, students, do you want to see the actual cell theory, which is not given in your NCERT, students? Do you want to see the actual cell theory, which is not part of NCERT, but it may be asked, but you should know sometime. Okay, students, do you want to see the actual cell theory, students? The actual cell theory here. This is called as fundamental features of cell theory. This right here is called as fundamental features of cell theory. See, this one is common here. All cells, all living organisms are composed of cells and the products here. Cells arise from pre-existing cells. These two are given in your NCERT. These two are given in your NCERT. Okay, NCERT. This next three, this is not there. Okay, there are only 5 of them. 1, 2, 3, 4, 5. Each cell is made up of small mass of protoplasm containing nucleus in its inside and a plasma membrane with or without cell wall on the outside. This is also part of your cell theory itself. All cells are basically alike in chemistry and physiology. Again, this is not true also to some extent. Okay, activities of organisms are sum total of activities of and interaction of their cell constituents. So, activity of a cell, activity for organism is basically sum total of all the activities of a cell. Now, this is called as fundamental features of the cell theory. Now, students, do you want to see modern cell theory? Students, see, modern cell theory is not given in NCERT at all. But do you want to have a look at it, students? Do you want to have a look at it? All of you? Can I get a fire emoji in the chat to let me know, sir, show us the modern cell theory? Because you'll be shocked. See, you'll be shocked because NCERT says, according to NCERT, you should only study these two. These two are enough, NCERT. This 3 are actually part of your cell theory, but not given NCERT. Okay, not given NCERT. But do you actually want to see the modern cell theory? One look you will give. Students, one look you will give. Students, one look. Here. I think all of you can't even read it. But if you can't read it, don't worry. I will give you this in PDF. In Telegram channel, I'll give you it as a PDF. So you can download from there the notes and read once. They will not ask you this, but you can have a look at it and read it once. Okay. Namrata ma'am class. Why Namrata ma'am? Namrata ma'am will do in J, right? Okay. So this is what is your actual cell theory, students. Crazy, no? Crazy. Now, students, is there any organism which do you think is not a part of cell theory? Anyone, students? Anyone? Okay. Any organism which is like, I will not be part of cell theory. Okay. There is something called as virus. Okay. Students, virus is actually not a part of cell theory. It does not follow. A virus is like, I'm my life, my rules. Virus is like, my life, my rules. I will only care for myself. So virus is like, cell theory doesn't apply to me because I am acellular. So viruses are, students, viruses are acellular or non-cellular in nature. Non-cellular in nature. They do not have cells only. Non-cellular viruses. Okay. Now, students, now with that we come to the end of the cell theory, students. Any doubt till now, students? Any doubt till now? We are going as detailed as possible. Okay. Any doubt till now, students? Anyone? Any doubt till now? Anyone? Any doubt till now, students? If there is no doubt, now we go to cell overview. Now we go to cell overview right now. Overview. Okay. Shall we start? Students, before I go forward, I want to know more about all of you. So, students, I want all of you to drop one word in the chat. Where are you from? All of you. Where are you from? All of you. I want to know. I want to see my new students. Where are all the students from? Quickly, drop a message, students. Quickly drop in the chat. Where are all you from? Which part of the country? Or outside India also? Who knows? Students, we never know. Okay, outside India also, we might be there. Someone might be there from this America or Europe or somewhere. In European country, anywhere. Yes, all of you quickly. Hyderabad, Tamil Nadu. Students, I can see one majority here. One majority is here. Okay, all Thambis in the chat. Ishwarya, if it is does not have a cell, I'll teach you, students. That ma'am will teach you in your bio classification, because viruses can become living once they infect. They become living once they infect. Okay, so all Thambis in the chat. Nice. APA, nice, students. Northeast, perfect, students. Nice. Now, students, will go over cell overview right now. Basic overview of the cell. Basic overview, students. Basic overview of the cell. Okay. Basic overview of the cell. Now, till now we have understood there is something called as unicellular and multicellular organisms. There is something called as unicellular as well as multicellular organisms. This we have learned right now. Students. Now, all of you should know there is something called as plant cell and animal cell also. Plant cell as well as animal cell. Both are different. Can anyone tell me one difference between plant cell and animal cell? Anyone? One difference between plant cell and animal cell? Anyone? Okay. Now, all of you should know there is something called as your plant cell, animal cell. Okay. Now, if you talk about your animal cell in general, students, yes, because yes, Jeshwit, your RBC and sieve cells, they are little exception to cell theory because they don't have nucleus. That RBC and sieve cells do not have nucleus only. Then there is something called as rhizopus also. That's a little exception here. Then there is cyanobacteria and monera is exception again, because they're made up of single cell. So there are multiple exceptions to modern cell theory. Okay. Now, students. Now, in the case of plant cell, we have cell wall. Animal cell, we do not have cell wall. Okay. That basic thing all of you should know. Now, all of you should know, students, there is something called as nucleus. Inside this nucleus, students, what do we find? Inside the nucleus, we find the genetic material. Genetic material can be in the form of chromatin or chromosome. That is basically the DNA here. Chromatin in the interface, and chromosome where? Chromosome in the M phase. All of you know that. Okay. If you do not know, don't worry, I will teach you cell cycle. I will teach you cell cycle, what is interphase, what is M phase, G1S, G2, pro, meta, anatilo. I'll teach you all that. Don't worry for that. It'll come up. Okay. Now, students, I want all of you to understand something right now. The overview. Okay. Overview of the cell. Okay. Overview of the cell right now. Okay. Now, okay. Let's talk about, first of all, let's divide into 3 parts. Okay. Last one doesn't require much actually. Okay. Okay. Now, we have, broadly speaking, we have 2 types of cells. Okay. We have something called as prokaryotic cell, and we have something called as eukaryotic cell. Okay. Eukaryotic cell. Okay. Prokaryotic and eukaryotic cell. Okay. Then there are some features which are common in pro and eu. There are some common features in pro and eu. Okay. Now, students. Okay. Fresher, yes. This is for fresher only, students. This is for fresher only. No, I'm going as slow as possible for a fresher itself. Okay. If you do not know what is chromatin, what is chromosome, don't worry. I will teach you all of that in cell cycle. Okay. Now, students. Now, I want all of you to understand one simple thing. In prokaryote and eukaryote, what is the difference here? All the prokaryotic cell, students, they will have something called as, in prokaryotic cell, students, there is no membrane bound organelles. All the organelles here, right? Or there is no organelles. In fact, there are not many organelles here. They do not have any membrane itself. They do not have any membrane at all. So here, no membrane bound organelles here in the case of prokaryotes. That is, they do not have an entire layer only, membrane. Okay. They have cell membrane on the outside, but apart from that, inside, nothing. No membrane bound organelles. But in the case of eukaryotes, they have something called as membrane bound organelles. Membrane bound organelles are present. They are present here, students. They are present here right now. Membrane bound organelles are present here. Okay. Now, students, now, in the case of your prokaryotes, they do not have any nucleus. No nucleus is not present only. So, if there is no nucleus only, the genetic material is scattered everywhere. Okay. Genetic material is scattered here. Now, here we have nucleus. Here we can find nucleus and everything. We can find nucleus. We can find here ER. We can find Golgi complex. All of them can be found here, students. All of them can be found here. Okay. Now, students, I want all of you to tell me, do you find any type of organelle in the eukaryote which does not have any membrane? Anyone, students? Okay. There are actually 2 of them here. 2 of them are such. There are 2 organelles here which do not have any membrane. Okay. The 2 organelles here are called as, students, one is your ribosomes, other one is centrosome. Ribosome and centrosome, students. Ribosome and centrosomes do not have any membrane around them. There is no membrane. And this ribosome, students, all of you know, ribosome helps in what? Ribosome helps in protein synthesis, while your centrosome, students, centrosome helps in what? Centrosome helps in cell division. It helps in cell division. Now, let me ask all of you one more thing, students. Do you find ribosomes here also? Yes, we do. Yes, we do, students. We find ribosomes here also. Ribosomes here also. Because non-membrane bound, right? Non-membrane bound. So we find ribosomes here. Now, students, but there is a small difference here. Small difference. The ribosome here and ribosome here are 2 different things. They're not same at all. Not same. The ribosome here is called as 70S ribosome. So don't worry if you do not know what is 70S. It is 70S ribosome here. And here we have 80s ribosome. 70s and 80s ribosome. Okay, that is the main difference. Now I want all of you tell me, but all these differences are there, but do you think there are any similarities? Yes, there is. The first similarity, students, both of them have cell membrane. Yes or no? Both of them have cell membrane. Pro and you. Both of them have something called as semi-fluid matrix. Can anyone tell me what is that? Semi-fluid matrix. Anyone? The semi-fluid matrix I am talking about is called as cytoplasm. Cytoplasm. So both of them have cytoplasm. And this cytoplasm, student, this cytoplasm right here, this is the ground for all the activities or enzyme reactions. All the reactions will happen in the cytoplasm. Okay, and this same cytoplasm can provide living state to the cell. Living state to the cell. Okay, that is your cytoplasm, students. Then the common thing is, again, there is DNA in both, genetic material. Then there is also something called as the ribosome is common, obviously. Ribosomes are common, right, students? So I will just move aside. All of you look at the notes and just try to understand. Any doubt you can shoot. Any doubt you can shoot, students. Any doubt here you can tell me, students. If there is no doubt, a quick fire emoji in the chat, because I want to know. My eyesight is a little weak here. Eyesight. I can't see the chat very easily, students. So any doubt here, students? Any doubt here? This is called as the overview of the cell. This is called as what, students? Overview of the cell. This is the entire overview of the cell. Okay. Done. Done, students. Perfect, students. Perfect, all of you are crazy. Now, students. Now, this is called overview. Now we talk about cell size. Students, let me ask all of you one simple logical question. Okay. When I say a small animal and a large animal, okay, do you think large animal has big, right? Large animal, students, do you think the large animal has bigger cell than compared to a small animal? Or the difference is that small animal has less cells, big animal has more cells? What is the difference, students? What is the difference? Main difference? I'll teach you mesosome. Don't worry, mesosome is coming. Okay. The main difference is the number of cells. Please remember that. Okay. But size of the cell also varies. For example, okay, the first thing is the smallest cell. Let's talk about the smallest cell, students. Let's talk about the smallest cell. Smallest cell. Anyone? Then we have the largest cell. Then we have the bacteria. Size we have to tell. Bacterial cell. Then we have the, let's say, human RBC. Then longest cell. Anyone, students? Anyone? You can start filling. You can start filling the. The smallest cell is actually part of kingdom Monera. The smallest cell is actually the part of kingdom Monera, which you might have learned yesterday. It does not have cell wall. It can survive in without oxygen, called as mycoplasma. Called as mycoplasma. Mycoplasma, students. Mycoplasma. And this mycoplasma, right, students, is anything between 0.1 micrometer to 0.3 micrometer. Very tiny. Like, can't even see. Obviously, you can't see, but it is so tiny compared to even a bacteria also. Obviously. Okay. Now, largest cell, student. Largest cell. Anyone? Largest cell is going to be ostrich egg. Ostrich egg. Ostrich egg. Now, students, bacterial cell. Anyone, students? Size of bacterial cell. Anyone? Size of bacterial cell. Anyone? Okay, I'll give you a small clue. Okay. It is less than 10 micrometer. It is somewhere between 3 micrometer to 5 micrometer. Students, all of you know how much is 1 micrometer? 1 micrometer is how much, students? 1 micrometer is 10 to the power of minus 6. Remember that. Okay. Now, what about RBC, students? 7 micrometer. 7 micrometer. Okay. Now, what about the longest, students? What about the longest? Longest is going to be nerve cell. Longest is going to be nerve cell. Okay, nerve cell. Nerve cell. Done, students? Done. Now, you can see, students, here. Nerve cell, red blood cells, white blood cells, columnar epithelial cells here, mesophyll cells, tracheids here. Okay. Now, can anyone tell me, students? Anyone here? Right? This tracheids are part of xylem of phloem. If you know, you know. If you do not know, don't worry. Tracheids are part of xylem of phloem in the chat. Tracheids are part of xylem of phloem. Okay. Now, students, today's main topic for today. After this, I will leave you. Okay. Today's main topic is called as prokaryotic cell. So we'll finish prokaryotic cell full today. Full entire class is going to be prokaryotic cell today, students. Ready, all of you? Ready? Shall we start, all of you? Shall we start? The first question for all of students: prokaryotic cell is observed in which of the following organisms? The first question to all of you: prokaryotic cell is found in which of the following organisms, or which kingdom? Just tell me which kingdom, students. Prokaryotic cell is found in your kingdom Monera. Kingdom Monera. Okay. Now, under kingdom Monera, obviously all of you know there is something called as your bacteria. Then there is BGA. Students, what is the other name of BGA, students? What is the other name of BGA in the chat? Then we have the mycoplasma, and we have the PPLO. PPLO. Fluoroorganisms. PPLO, students here. Okay. BGA is also called as your blue-green algae, is also called as cyanobacteria. Also called as what? Cyanobacteria here. Okay. Now, students, I want all of you to understand this. Yesterday, Gopika ma'am would have told all of you. Gopika ma'am would have told all of you, students. There is something called a shape of cells. How many of you know there are different shapes? See here. There are different shapes here. There is cocci, there is bacillus, there is vibrio, and there is spiral shape. Cocci is spherical. Cocci is spherical here. Spherical here. Bacillus is rod shape here. Then we have vibrio is comma shape. Spirilla is the spiral shape here. Spiral shape. Okay. Now, let me ask you all of you one simple question right now. When I say a prokaryotic cell, what is coming to your mind right now, all of you? When I say a prokaryotic cell, what is coming to your mind right now, students? All of you, quickly smash the like button. First of all of you, quickly smash the like button right now and tell me the answer. When I say a prokaryotic cell, what is coming to your mind right now, students? Prokaryotic cell. What is coming to your mind right now? Bacteria? Yes or no? Tell me, students. Tell me what is coming to your mind right now. When I say a prokaryotic cell, what is coming to your mind? This or no, student? This me? Tell me. Do you think this image is coming to your mind right now? Is this image coming to your mind right now, student? This right here is a prokaryotic cell. Prokaryotic cell, right? A prokaryotic cell, students, a prokaryotic cell will have a flagella here. Flagella is seen here. The students, do you see here? It has your 3 layers outside. Then it has ribosomes on the inside. It has genetic material. Genetic material is completely scattered here. Genetic material, students, is completely scattered here, called as nucleoid. Nucleoid here. Nucleoid here. This right here is your, this right here is your prokaryotic cell. Now, students, I want all of you to quickly run and get a book. All of you. I want all of you to quickly run and get a book, because now I'll make you write about the entire structure. I'll make you draw the entire structure. If you do this right now, the chapter, the prokaryotic cell is over. Okay. So quickly run, get a book, all of you. Once you get a book, just say done sir in the chat. All of you. You're listening to a podcast. Don't listen to a podcast. Go get a book fast. All of you. Quickly run and get a book. Okay. Now, once you get a book, students, I want all of you to start drawing here. I want all of you to start drawing right now. Okay. I want all of you to start drawing. Okay. Start drawing, students. Students, can all of you see here? There are 3 different layers here. Students, 1 second, students. My dog is going mental here. I think someone came home. Okay. Friends, can all of you see 3 different layers here, students? Yes, got the book? I want all of you to draw 3 different structures like this. Now, this right here, students, this right here is a typical prokaryotic cell. This is a prokaryotic cell, student. This is a prokaryotic cell. Now, I want all of you to label. I want all of you to start labeling one by one. Okay. Now, in this prokaryotic cell, the outermost layer is known as what? Here, this one. The outermost layer, students, here, the outermost layer is called as glycocalyx here. The outermost layer is called as glycocalyx. Okay, glycocalyx. Now, this outermost layer, glycocalyx, can be found as a, let's say, as a loose sheath, or it can be found as thick and tough layer. Thick and tough layer. Okay, glycocalyx. Glycocalyx, students. Now, when it is present as loose sheath, it is called as what? It is called as loose sheet, called as slime layer. Slime layer. Loose sheet is called as slime layer. Okay. Now, when it is present as thick and tough, it is called as what? It is called as capsule. It is called as capsule. That is the outermost, students. That is the outermost layer. Okay. So we have here slime layer, and capsule here. Glycocalyx is the outermost layer of your entire prokaryotic cell. Done, students? Done. Now I want all of you to understand which is the second layer right now. The second layer is called as cell wall here. Second layer is called as cell wall here. Now, what is cell wall made up of? The cell wall is made up of something called as peptidoglycan. The cell wall is made up of peptidoglycan. Peptidoglycan. Okay, peptidoglycan. Now you'll ask me, sir, what is peptidoglycan made up of? Peptidoglycan, students, is made up of something called as NAG and NAM. Peptidoglycan is actually made up of something called as NAG and NAM. NAG and NAM. Students, do you want to see the structure of peptidoglycan, students? Do you want to see the structure of peptidoglycan? Yes or no, students? Let me check quickly. All of you want to see, students? Yes or no? This right here is a peptidoglycan. Can you see, students? This right here is a peptidoglycan. It is made up of NAG and NAM. N-acetyl glucosamine and N-acetyl muramic acid. This is your NAG and NAM. Okay. Here we have NAG, here we have NAM. Repeating units, NAG and NAM. Along, students, along with that, we also have different types of amino acids here. Different types of amino acids are also present here. Okay. Alanine, glutamic acid, lysine, alanine, everything is present here. This is how the peptidoglycan looks like. This is how the peptidoglycan looks like. Okay, it is made up of NAG and NAM. Now, which is the innermost layer, students? The innermost layer is called as the cell membrane. It is the cell membrane, students. Innermost layer is called as cell membrane. Now, this innermost layer, cell membrane, students, all of you should know it is very selectively permeable. Selectively permeable. It will not allow anyone inside. This cell membrane will not allow any random person to enter inside. Not possible at all. So here we have, students, the entire prokaryotic cell has 3 main layers here. First, we have the entire thing is called as glycocalyx. Glycocalyx can be in the form of 2 forms: loose sheet or called as slime layer, thick and tough called as capsule. Then we have the cell wall here made up of peptidoglycan, which is basically NAG and NAM and certain amino acids. Then we have cell membrane, which is selectively permeable in nature, will not allow anyone randomly inside. Okay. Now students, what do you have on the inside? On the inside, the main is the cytoplasm. It is the cytoplasm on the inside, students. So okay, we have the cytoplasm on the inside. Let's say we draw the cytoplasm here. Cytoplasm. Done? Done, students. Done. Now I want all of you to understand, students, this particular structure called as your cell membrane or plasma membrane will have certain something called as extensions. Let me just show you, students. Let me just show you. Just take a paper and show you, students. Okay. So let's say this is a cell membrane here. This cell membrane will have certain, extension like this, extensions. These extensions of cell membrane are called as mesosomes. Okay. Let's draw extension here, students. Now this is the cell membrane. It will have extension like this. Let's say extension. Okay. This extension of cell membrane. Let me just do color filling here, students. Color. This is called as extension of cell membrane, students. And this extension of cell membrane is called as mesosomes. Mesosomes. Okay, that is called as your mesosome, students. Done, students. This mesosome has a lot of function. That function I'll teach you in the next slide. It has DNA replication, respiration, cell wall formation, increasing the surface area. All that is there. I'll teach you one by one, students. I'll teach you one by one. That is called as your mesosomes. Now, similar to mesosome, there is one more extension in the cell membrane here. Similar to mesosome, there is one more extension here. Can all of you students? There is one more extension here. Students, am I audible, right? Okay. Students, am I audible to all of you? Okay. Similar to mesosome, there is one more extension here. Can you see extension? Here. This extension. Now, students, this is one more extension here. Now, this extension is called as chromatophore. Okay. This extension is called as chromatophore. Now, what is the main function, students? This chromatophore actually has pigment in it. And this chromatophore is found in your BGA, blue-green algae. So, chromatophore can be found in your blue-green algae. Okay. Blue-green algae. Okay. Done, students. Now, students, I want all of you to understand from the cell wall, there can be a structure like this. Extension of cell wall. Okay. From the entire cell wall here, there can be extension like this, which helps in motility. Can anyone tell me what is the structure here? This extension of cell wall here, this extension of cell wall, this structure is called as flagella. This structure is called as flagella. It's called as flagella. Flagella helps in movement and motility here. Now, similar to the flagella, there is something also called as, students, a tube-like structure here. Okay, tube-like structure here, students. This tube-like structure here is called as pilae or fimbriae. It is called as pilae or fimbriae. Okay. Now, all of you told me this main function of flagella is motility. Then what is the main function of pilae and fimbriae? The main function of pilae and fimbriae is not motility, it is actually attachment. Attachment to substratum. Okay, attachment to substrate. Okay, pilae, students, pilae helps in conjugation. Mainly in conjugation. Helps in conjugation here. It helps in conjugation. That is pilae and fimbriae. What is conjugation, students? Conjugation is basically a type of your type of reproduction here, where the exchange of genetic material happens. This is one cell, one more cell. Between the two cells, there is a bridge formation. That is called as the entire pilae formation. Okay, conjugation formation. Okay, now pilae and fimbriae then. Now, students, anything outside we have left? No. Outside we have covered everything. Okay. Flagella, Pylian fimbriae, attachment, chromatophore pigment in blue-green algae, mesosomes is the extension of cell membrane here. Done. Now, what do we have on the inside, students? On the inside, do we have the genetic material here? Yes, we do. But do you think the genetic material is free floating or inside the nucleus? Students, do you find a nucleus here? All of you, do you find a nucleus, students, here? Do you find a nucleus here, students? Anyone? Yes, ask the doubts. Ask the doubts, students, ask the doubts. Do you find a nucleus here? No, you do not. There is no nucleus here, students. All the genetic material is present as such. Genetic material is present as such. And this structure here, genetic material that is present as such, is called as nucleoid. It is called as nucleoid here. Okay, nucleoid structure here. Now, students, apart from nucleoid, do they have ribosomes here? Yes, there is ribosome here. Okay, large subunit, small subunit. This ribosome is called as 70S ribosome. It is a 70S ribosome present here. 70S ribosome here. Now, apart from the main genetic material, which is basically your double-stranded DNA here, okay, which can be circular or straight, which can be circular or straight. This is your double-stranded DNA. Double-stranded DNA, which can be circular or straight. Circular or straight here. That is your DNA, double-stranded DNA here. Apart from the students, do you think there is extra chromosomal DNA here? Somewhat circular, self-replicating? Yes, there is. There is something called as plasmid here. There is something called as a plasmid also here. Okay, something called as plasmid. This is extra chromosomal self-replicating DNA. Extra chromosomal self-replicating DNA called as your plasmid also is here, students. Apart from this, do you think anything is missing here? Yes, there is. There is something called as inclusion bodies. Inclusion bodies are like storage. Like we have vacuoles, right? Vacuoles. Similar to vacuoles, there is something called as inclusion bodies also. Okay. Where do we write? Where do we write? Okay, we write here only, students. Okay. There are inclusion bodies here. Inside a cell, there is something called as inclusion bodies. Round structures. Storage. For storage. Okay. These are called as what? Inclusion bodies. Inclusion bodies are also present here, students. Now, how do you think the entire concept, students? How do you think the entire slide? I'm going to move aside. Just try to take a screenshot right now and just try to look at it. How beautiful it looks. All of you students. How beautiful is it looking, students? Tell me in the chat. Students, how beautiful does it look? Tell me, students, in the chat quickly. How beautiful does it look? Crazy, no? All of you. All of you students? Yes or no? There is no mitochondria here. There is no plastid here. There is no mitochondria, no plastid, nothing. All of you students. Now, if you open your NCERT, students, okay, let's just finish the entire thing. Okay, let me just finish the entire thing and then I'll show you NCERT. Okay, students, let me just look at it. This is what was given here. This is what we drew. I hope all of you drew with me, students. I hope all of you drew this with me. Like, this is what we love in biology, the colorful, diagrams and everything in biology, where we cover the entire board, right? Beautiful, right, students? Beautiful? Yes or no? Crazy. So I hope all of you also drew very nicely in your book also. Students, did all of you draw very well? All of you drew also very well, students? Yes or no? So students, I know you are confused. So students, do you want me to give a small recap of the entire thing? Do you want me to give you the small recap of the entire structure here, one small recap? Okay. Now listen to me, students, listen to me very carefully. Listen to me very, very carefully right now. One small recap of the entire thing. Okay, small recap of the entire thing, students. Once again, one small recap of the entire thing. Okay, first of all, students, a prokaryotic cell does not have any membrane-bound organelles. Doesn't have any membrane-bound organelles are not present at all. Okay, it has how many layers? Can anyone tell me, students, how many layers are present here? It has 3 main layers. The outermost layer is called as glycocalyx, which can be present in loose sheath called as slime layer, thick and tough called as capsule. Now second layer is called as cell wall, made up of peptidoglycan, NAG and NAM. Okay, N-acetyl muramic acid, N-acetyl glucosamine. Okay, that is NAG and NAM. Then innermost layer is called as cell membrane, which is selectively permeable in nature. Selectively permeable in nature, students. Okay, now then we have a flagella here, which helps in motility. Then we have pili and fimbriae, which main function is attachment to the substratum here. Okay, pili helps in conjugation also. Then in the genetic material is DNA, circular single straight here, double-stranded DNA called as nucleoid here. Then you have cytoplasm on the inside. Then in the cell membrane there are extensions here called as mesosomes, extensions of cell membrane. Then along with the extension of cell membrane, we have something called as chromatophore also here. Chromatophore is basically an extension of cell membrane, but it has pigment inside. It has pigment on the inside. Okay, and it is present in your blue-green algae here. Okay, then we have ribosome, that is 70s. Then we have inclusion body also. Then we also have the inclusion body here. Inclusion body, students. Now next main thing is, students, all of you need to understand, which they can ask you in the examination, is what is mesosome and what is the function of mesosome. Okay, so what is mesosome is what? Mesosome is extension of cell membrane. Yes, it is the extension of cell membrane. All of you know that. Now this mesosome can be present in 3 different forms also. It can be like, vesicle, round on. It can be tubular, or it can be lamellae shape. Or it can be lamellae shape also. Now, what are the main functions of mesosomes? Understand. Very important. What are the functions? This is where the questions will come from. Okay. Functions of mesosomes. Understand all of you. The first main function is called as your DNA replication. Then you have cell wall formation. Okay, then we students have the helps in respiration. It acts like a mitochondria. The mesosomes act like a mitochondria, so they help in respiration also, students. Now all of you students, every single time you want to wipe something, you increase the surface area. So can I tell mesosomes also increase the surface area because it is forming the foldings here? Yes, it does. So it increases the surface area. Then students, it helps in distribution of daughter cells. That is basically, it helps in replication also. It helps in cell division also. Students. Okay. Apart from that, it can also be secretory in nature. Students, be secretory in nature. Also helps in secretion. Okay. Apart from all, students, it also has a lot of enzymes also. That is your mesosomes here. Now then there is, students, something called as gram-positive and gram-negative bacteria. Okay. This is basically a type of classification of bacteria. And if you can see, students, there is something called as in the case of gram-positive and gram-negative bacteria, this is the main difference. In gram-positive, we have more peptidoglycan. Gram-negative, very less peptidoglycan here. That is the main difference between gram-positive and gram-negative bacteria, students. Okay. Gram-positive and gram-negative bacteria. Okay. Function of inclusion body is coming up, don't worry. Inclusion body is coming up. Okay. Done, students? Done? Okay. Now, all of you need to understand the flagella here, students. Flagella. Students, do you want to see an animation of flagella? How does the flagella actually works here? It is like a motor. Here. Okay. This flagella, students, actually has three main parts here. Okay, flagella, whose main function is motility here. It has three main parts here. Three main parts. Okay, three main parts here. The first main part, bottom part, is called as basal body. It is called as basal body. Then it has a hook. Then it has the long flagella, filament here. Long filament. Okay, this is basically the flagella here. Okay, this is basically the flagella, students, here. Flagella. Okay, one second, students, one second. Let me just show you the flagella working. It looks like a machine. Looks like a machine. It looks like a machine, students. Looks like a machine. This is how it looks, students. Can all of you see? It looks like a machine. Can you see? It has, this is the basal body. This is the entire thing called a hook. Then you have a filament. It will keep on. They have rings. There are multiple rings here. 3 rings, 4 rings, every present. All the rings will be rotating here. And this is the lipid bilayer. Can you see lipid bilayer? Cell membrane here. Peptidoglycan here. Okay, peptidoglycan here, students. This is how it rotates. Okay, this is the flagella looks like. This is how the flagella looks like, students. Okay. Now, next, this can you see here? This is the basal body, hook and filament here. This is the entire basal body part which rotates here. Okay. Now, let's understand ribosomes here, students. What is the type of ribosomes you see here? Now, all of you should know what is the main function of ribosome. Do they help in transcription or translation? They help in translation. And ribosomes, right? They help in protein production. Now, students. Now, a typical ribosome is, in this case, is 70S. 70S is divided into 50S and 30S. Here, this is called as your large subunit, and this is called as small subunit. Okay. Now, students, very important concept here. Large subunit also has multiple parts. Small subunit also has multiple parts. Now, one question arises. Okay, what is it made up of? Students? What is actually ribosomes made up of? Ribosomes are actually made up of RNA and proteins. Okay, ribosomes are made up of what, students? They're made up of RNA plus proteins. Okay, they made up RNA proteins, and they will help in production of proteins. This is hilarious. You'll get confused otherwise. They help in production of proteins. They're made up of RNA and protein. So all of you can see here, students, 50S is made up of what? 23S, 5S rRNA and 34 proteins. 16S rRNA and 21 proteins. So it is made up of what? The entire ribosomes are made up of, again, they're made up of RNA plus proteins. That is what they're made up of. That is what the ribosomes are made up of. Now, some of you will be getting confused right now, students. Okay. Now, right. Some of you will be like, sir, how is 70S equals 50 plus 30? Yes or no, students? Is that confusion is there? All of you try to add 50 plus 30 and tell me the answer. All of you try to add 50 plus 30 and tell me the answer in the chat, students. All of you add. I want all of you to add 50 plus 30 and tell the answer in the chat. All of you. All of you, students. What is 50 plus 30 in the chat, students? 50 plus 30 is how much? 50 plus 30 is how much? How much 50 plus 30? Okay. Okay. 80. But here I am telling 70 here. Why? Because, students, understand, this right here is not mathematical. This is biology. This entire thing is based on sedimentation coefficient. Okay. This actually what happens is, this is a small experiment done. In a small experiment, what do? We take a test tube here. In this test tube, in this entire test tube, we add your 70S, we add 50S, and we add 30S here. 30S. Okay. And this test tube undergoes centrifugation. Now, based on density, they will separate out. So this number which you obtain here, 30, 50, 70, this is based on the sedimentation coefficient and density of the structure here or the organ here. Okay, so they can move around. So 30 will somewhere here, 50 is somewhere here, and 70 is somewhere here. So this is based on sedimentation, not on mathematical. So when you add 50 years, 30 years, 70 years in a test tube and rotate very fast, they will separate out. This is how they separate out here. This is how. So this is based on sedimentation coefficient, not based on the number here. Okay. So students, oh my god, what did I do? Okay. So here, some of you will be like, sir, what exactly is this S here, students? In this 70S, what is this S? The S is called, student, the S is called as Sedgwick unit. Sedgwick's unit. Sedgwick's unit right now. Also, the S is also called as sedimentation coefficient. Sedimentation coefficient. Sedimentation coefficient. That is your S. Sedgwick or sedimentation coefficient. 70S. Okay. 70S, students. Okay. Sedimentation coefficient. That is your S basically here. That is your S here. Sedimentation coefficient. Okay. Now, next concept is called as polyribosome. What is polyribosome, students? First of all, students, tell me in the chat, what do you mean by poly? Poly means one or many? Poly means many. Poly means many. Okay. So what is happening in polyribosome is that there is a single large mRNA, a single mRNA, right, plus many ribosomes. Many ribosomes. Okay. So on this mRNA right now, we can find multiple ribosomes here. Multiple ribosomes on the single mRNA. That is called as, students, that is called as what? That is called as polyribosome. Polyribosomes or polysome. Polysomes. Okay. It is called as polyribosome or polysome. Okay. Polysome. Polysome. That is basically polyribosome or polysome. That's all, students. Now, under prokaryotes, this is the last concept, students here. Last concept called inclusion bodies. Inclusion bodies are like dustbins or storage units. Okay. Inclusion bodies, students, are like your storage unit. Students, how many of you have that almara in your house? One bureau in your house, students? Bureau you have? All of you in the house where you keep, inside you keep gold also, you keep saris, mother will keep all the sari, everything inside. That is like your almari here, almari or bureau. That is your inclusion bodies in the case of your prokaryotic cell. Inside this prokaryotic cell, you can keep all the different items here. All different items. Okay. Now there are different types of inclusion bodies here. Different types of inclusion bodies. For example, there is something called as phosphate granules. Phosphate granules. Students, then there is something called as Cyanophesian granules. Cyanophesian granules. Okay. Then there is something called as your glycogen granules. Okay. Glycogen granules are there. Apart from this, student, there is something called as gas vacuoles also. Gas vacuoles. The interesting part is, students, these gas vacuoles are only found in your blue-green and purple sulfur bacteria. Okay. Or let's say photosynthetic bacteria. Photosynthetic bacteria. Now you'll ask me, sir, what is the main function of the gas vacuoles? Okay, gas vacuoles will help the bacteria to buoyancy. Now all of you know what is buoyancy. You take a water bottle, throw it in, take a water bottle with gas in it, you throw it in the water. The bottle will float on top. Bottle will float. Why? Because the bottle has gas in it and it can float on top very well because of buoyancy. So similarly, students, because of buoyancy, here also the gas, because of gas vacuoles, the bacteria can float on top. Okay, float on top, students. Students, with that, entire prokaryotic cell is over. Here is the question. Quickly answer in the chat, students. Quick answer in the chat, students. Quick answer in the chat. Buoyancy. Yes. Students, answer in the chat quickly, all of you. Answer in the chat. All of you, quickly. Done. Cell wall formation is mesosomes. Mesosomes. Next question on the board. Next question on the board, students. Quickly, students, quickly. What is the answer, students? What is the answer? Quickly. Gas vacuoles. Next question. Concept of omnicellular E-cellular was given by. Concept of omnicellular E-cellular was given by. Concept of omnicellular E-cellular was given by. Radolf Virchow. Radolf Virchow, students. Radolf Virchow. Okay. Now, students, let's do a NCERT recap. Let's do a NCERT recap right now, students. NCERT recap. Okay. Students, is the board visible to all of you? Is the board visible to all of you? Yes. Yes or no? Is the board visible to all of you, students? Yes. 15 minutes recap right now, students. Once again, we'll see. 15 minutes recap. Okay. 10 minutes recap, all of you, before we go. Okay. So, students, this is the introduction part of the cell chapter. This is the introduction part of cell chapter, students. Here, this also becomes very important to study once. Okay. So, biology is the study of living organisms, all of you know. Okay. The detailed description of their form and appearance only brought out their diversity. It is a cell theory that emphasizes the unity underlying this diversity of forms. That is, cellular organization of all life forms. A description of cell structure, cell growth by division is given in the chapter and comprising. Okay. Cell theory also created a sense of mystery among living phenomena also. Because cell theory, how is it coming, students? How is the cell theory even happening? All of you know right now. Cell theory given by two scientists, Scheiden and so on. Then we have Radolf Virchow, who modified the cell theory. Okay. Now, students, see this. Now, physiological and behavioral processes. This mystery was requirement for the integrity of the cellular organization of the living phenomena to be demonstrated or observed. In studying and understanding the physiology and behavioral process, physiological and behavioral process, one can take a physiochemical approach and use a cell-free system to investigate this. This approach enables us to describe the various processes in molecular biology. Okay, so if you use physical and biochemical approach, you can understand the chapter, you can understand the cell even better. They're saying. Okay, it will tell us what type of organic compounds are present in the living organisms. This we'll learn in the biomolecules chapter, student. This we learn in biomolecule chapter. So this physiochemical approach to study and understand living organisms, the physicochemical approach to understand an organism, right? Physicochemical approach to study and understand living organisms is called as reductionist biologist. This is a NEET PYQ from 2025 examination. Reductionist biologist. Okay, reductionist biologist, right here, student. Reductionist biologist. Okay, so what is reductionist biology, students? The physicochemical approach to study the living organisms is called a reduction biologist. Okay, now students, now Ramachandran. All of you need to know about Ramachandran. G. N. Ramachandran, students. Ramachandran was a scientist who was founder in the Madras School. So he's from your Tamil Nadu itself. Madras School of Conformational Analysis of Biopolymers. So if someone asks you what is the main function of your Ramachandran, the main function of Ramachandran students, is his detailed analysis on your protein structures, right? There is something called as Ramachandran plot, which we use to understand the protein structures and everything. Protein structures. Okay. So you can read all the students called as Ramachandran plot. Ramachandran plot. Okay. Then students, we understand the cells chapter here. We learned about what is cell. All organisms are composed of cells. Some are composed of single cell, that is called unicellular. Some are made of multiple cells, that is called multiple cellular right now. Multiple cellular, students. Made up of many cells called as multiple cellular. Then we know what is a cell. Yes, what is a cell. Then we learned about your Anthony von Leeuwenhoek, who saw the first living cell. Robert Brown discovered the nucleus later on, students. Nucleus later on. I told you unicellular organisms are capable of independent existence and performing all essential functions of life. Yes. Then we learned about cell theory. Matthias Scheiden, German botanist, who told about us plants. Right? Plants are made up of different kinds of cells and tissues. Then we learned about Theodor Swan right now. Right? Theodor Swan. He was a German zoologist right now. Okay. He studied different kinds of animal tissues. He told us that animal cells has a separate outer lining called a cell membrane. In the case of plant cell, there is something called a cell wall. See here, students. Then outer layer, today we known as plasma membrane, studies that presence of cell wall is unique to your plants here. Swan proposed the hypothesis that is animals and plants are composed of cells and product of cells. That is basically your cell theory. That is basically the cell theory, students. Okay. So Scheiden and Swan together formulated the cell theory. Okay. Now this theory, however, did not explain the new cells are formed. How new cells are formed. So that concept was given by Rudolf Virchow, omnicella e cella. He told us that new cells exist from a pre-existing cells. Okay, see here, students. Rudolf Virchow explained the cells divide and new cells are formed from pre-existing cells. Okay, this is your cell theory given here. Then we learned the overview of the cells. Prokaryotic cell, eukaryotic cell, everything is given here, students. Which I told you before about ribosomes and everything I told you, right? Everything is given right now here, students. Different type of cells done. Then prokaryotic cell, students. Prokaryotic cell. Here, see. Prokaryotic cells are basically your kingdom Monera, bacteria, blue-green algae, mycoplasma, PPLO, and PPLO right here. PPLO, students. Okay. Different shapes. Cocci, bacilli, spiral, and vibrio. Okay. Vibrio, students. Now, students. Here are the different shapes, sizes also given. Eukaryotic cell, viruses, PPLO, typical bacteria. Okay. Here it is written. Okay. Typical bacteria also. Okay. Now, students, here you can just read this one entire thing. It just talks about your how ribosome is common to both of them. Here it is telling you about how DNA genetic material. Now, cell envelope, you can see, students. Most eukaryotes, particularly bacteria, have chemically complex envelope. It has 3 layers. Outermost called as glycocalyx, followed by cell wall and plasma membrane on the inner side. Okay. Although each layer of the cell envelope performs distinct function, they act together as a unit here. Okay, unit. Gram positive and gram negative bacteria, difference in the peptidoglycan here. Okay. Gram positive will take up the crystal violet, gram negative will not take up the crystal violet. That is the stain, which we use in gram staining. Now, glycocalyx, slime layer, capsule. Slime layer 1, loose sheath, capsule, thick and tough. Thick and tough, students. The cell wall, right, prevents the bacterium from bursting or collapsing. Okay. Plasma membrane is selectively permeable. All of you know that. Selectively permeable in nature. Okay. Now, students, a special membrane structure called as mesosome. What is mesosome, students? Mesosome is the extension of cell membrane. That is called as mesosome. Functions, see here, students. It is in the form of vesicle, tubular, lamellae shape. I told you. Now, what are the functions? DNA replication, distribution of daughter cells. Yes. Secretion, respiration, everything. Enzyme content also. Okay. Now, students, similarly, there is one more extension. Similarly, there is one more extension of cell membrane called as chromatophore. All of you know what is chromatophore? See here, chromatophore. Right? It is present in the case of cyanobacteria. Chromatophore is present in the case of cyanobacteria, which contains pigment. Now, bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called as flagella. Yes, no, students? All of you understanding? So they have a thin filamentous extension from their cell wall here. Okay, called as flagella. Extension is happening. Bacterium shows a range of number of arrangement in flagella. It has 3 parts: filamentous, hook, and basal body. Okay, the flagella is the outermost part here. So filament is the longest portion extending from cell surface. From the cell surface to the outside wall. From the cell surface, it is extending to the outside. Okay. Now, beside flagella, pili and fimbriae. Pili and fimbriae will not help in motility. They help in attachment. They help in attachment, students. Okay. The pili are elongated tubular structures, right, made of special proteins. Fimbriae, small bristle-like. So fimbriae are like small bristle-like. The meaning of bristle, all of you know, toothbrush. All of you brush know, toothbrush. A toothbrush has the small finger, tiny structures that rather know in brush. That is called as bristles. Here also we have bristles here. Okay, they help in attachment. Then ribosome, students, ribosome. Then 50S, 30S ribosomes. What are they made up of, students? They're made up of RNA and they're made up of your proteins. Okay. Now, polyribosome means single mRNA and multiple types of proteins. Multiple types of ribosomes. Then we have inclusion bodies. Inclusion bodies is what? Reserved material in prokaryotic cells are stored in the cytoplasm of the inclusion bodies. Okay, they are free floating in the cytoplasm. Examples: phosphate granules, cyanophysian granules, glycogen granules, gas vacuoles, where blue-green and purple-green cells of bacteria, photosynthetic bacteria. With that, today's class is done, students. Today's class is over today. Thank you so much, students. With that, today's class is over. Okay, I thought I will finish some amount of eukaryotic cell also today, but I don't think it's possible. Eukaryotic cell, we'll do it in next class, part 2. Okay, so part 2 will be a little longer because part 2 has a lot of information. Okay, we should start from endomembrane system, mitochondria, chloroplast. Then we're supposed to do cytoskeleton structures, right? Ribosomes. It'll take a little bit of time. Okay, so students, that's it for today's class. Now all of you have one simple job, students. I'll give you a homework question. Tell me the answer in the chat. Okay, a simple homework question. Like a simple homework question. Simple homework question, students. I want all of you to name. Right, one organelle. Right. I want all of you to name one organelle which is common in prokaryote and eukaryote in comments. One common organelle, right? One common organelle which is common in prokaryotes and eukaryotes. Okay, all of you do that, students. All of you do that. Yes. Animal cell is cell membrane. Yes, it says cell surface, not cell membrane. It says cell surface. Not here, students. Not here. In the comment section. Not here. In the comments. I will not see. I will close my eyes now. I'll close my eyes. I will not see. Okay. I will not see. Put it in the comment section. Not here, students. Put it in comment section. Okay. Okay. Okay, students. In the comment section. Not here. Okay. Raha, Rastu, students. And also in the comment section, you should also add how was the class. How was the class also. Okay. So I will look forward to all the comments. Sumita, Renuka, Kadupalli, Sumiti, cute bunny, cute bunny. Okay. Harini, Nagarjuna, Varshita. Okay. Then Jashwit, yes, Rusty, Aishwarya. All your comments I will see in the comment section. Okay, students. So with that, today's class comes to an end, students. I hope all of you enjoyed today's class. Let me know in the comment section, students, how was the class, all of you. Okay. So until then, students, bye-bye. Take care, all of you, and have a great day ahead. Okay. All of you, students. Bye-bye. Okay. Bye-bye, students. Boy, all of you, boy. Students, before I go, one small thing which all of you should know, and you will be knowing by now, right? Right. All of you will be knowing right now, right? We have the DNA batch also. See, this is a free batch here. We do not give you like notes are given, but there is no quiz. There is no doubt solving much. In the DNA batch, we have one more teacher to solve your doubts, and I'll be teaching there. And I've already taught 2 chapters there. Okay, cell cycle and cell unit of life. So I'll be starting a new chapter called as biology classification there also. Okay, so this is your DNA batch. Link is not missing here. Okay, students here. See, DNA batch 2026. So if you're 2026 aspirant, DNA batch is for all of you students, right? It is at 499 right now. And here in DNA batch, you will get a doubt support. You will get Tattva books, soft copy. You will also get students test series. You will get unlimited DPPs, everything possible which you need to cracking NEET in this DNA batch. Let's say you are offline student. So in that case, what should you do? If you're an offline students, you should actually join our students. You have to fill this form. So if you are in Chennai, if you are in Chennai and near Annanagar, or if you are in, let's say, in Hyderabad, or if you're anywhere in Andhra Pradesh also, we have multiple offline centers which you can visit. So I want all of you to visit the offline center once and check it out once. Now, if you want to check the offline center, you can fill this form right now, students. You can fill this form here. It is not opening, but if you click here, it will open. Okay. So offline center, that is there. Okay, students. So that's it for today's class. Let me know in the comment section how was the class in the comment section, and I will look forward to it. Part 2 coming up in this week itself. No, next week, part 2 next week. Okay. So bye bye, students. All of you take care, and I'll see you in the next class. All of you, students. Bye bye. Students, price of the batch is given right here. All of you can see, students. Students, where did I go? Just go to live here. Students, tomorrow class is there. Please forget this class. Entire Q&A is there tomorrow for all of you to doubt clearing is there. Okay. So if you can go here, students. Here, if you go DNA batch, here you can go here. This is the price right now. Okay. It was 7,000. We decreased to 5,000 right now. This is the batch price right now, students. Okay. So bye bye, all of you. Take care and sayonara, all of you. Sayonara.